We will continue with the lesson series, A Tour Through a Cartoon Cell, and we'll focus this time on getting and using energy. Here's our cartoon cell, and we have the four categories of molecules that cells are composed of. In this lesson, we'll focus on the sugars like glucose, a principal energy molecule for many cells on planet Earth. In the previous lesson, we learned about two sets of chemical reactions, one to extract energy from food and the other to build molecules. These together form what we call metabolism, the set of chemical reactions that keep cells alive. In this first lesson, we'll focus on the reactions that get energy from materials in the environment, like glucose, and then we'll take a look at building projects. So in previous lessons, we've learned that the molecule glucose has energy locked up in all these chemical bonds. Photosynthesis used the energy of light to make the chemical bonds in glucose. As a result, glucose stores potential energy in its chemical bonds. So therefore, breaking these chemical bonds will release the stored energy. So that's going to be the approach that cells take. If they can get this molecule, then if they break these chemical bonds, they can liberate the energy that was stored in those bonds. And that's going to be the scene right up here at the top of the cell. Glucose is being transported across the membrane by a channel protein, and now we're going to have a team of proteins begin the process of glucose breakdown. So specialized proteins uniquely suited for this task begin a chemical breakdown of the food molecule. So here we'll look at this purple protein here, and it's going to, in a sense, attack. Now that's an aggressive description, but basically glucose is going to bind to the protein, and the protein is going to break a chemical bond. And when a chemical bond is broken, energy is released. Now, the energy doesn't just dissipate as heat. The cell has ways of harnessing this energy and transferring that energy to another molecule, as we'll see in a moment. We should remember that proteins are chains of amino acids that fold up into a three-dimensional shape, and the shape of the protein determines what it can do. And in a previous lesson, we saw this protein here. It's really myoglobin, but we're going to pretend that this is the protein that's going to be attacking glucose and breaking down glucose. But just to remind ourselves, the proteins are very large molecules. So if we pretend this is the protein involved in glucose metabolism, the idea is it has a binding site, a sticky place where glucose can stick to it. And that's the active site of the protein. And now the protein is getting ready to break one of its chemical bonds. And so there, the chemical bond has been broken, and energy as a consequence is released from this. So the protein has done its job. It has begun the metabolism of glucose to extract energy from the molecule. So let's return to our scene up here. Here we have this first interaction. Glucose is going to stick to this blue protein, and the blue protein will break one of the chemical bonds of glucose. But that's not the end of the process. There's a lot more chemical bonds in glucose to be broken. Well, that's where the next protein and the next one after that comes into play. So there is a whole team of proteins here whose job is to break many of the chemical bonds of glucose. But it's the blue one that has to go first. Then the resulting molecule will be floating around, and if it encounters the green one, then the green one can break the next chemical bond, and so on. So there is a whole team of these proteins whose job it is to begin the breakdown of glucose. Now, these proteins do not have eyeballs. They don't know where to find glucose. Rather, all of these molecules are simply bouncing around within the fluid cytoplasm of the cell. In fact, the way the cell speeds up these chemical reactions is to make many of the relevant proteins and to import as many of the nutrient molecules, as many glucose molecules as possible. So the more proteins the cell makes and the more glucose molecules that are in the cell, then the chances of a random collision increase. So the cell can achieve an important chemical reaction by just making enough of the relevant proteins and having enough of the target molecules so that random interactions will produce the desired outcome. In this case, the first chemical bond of glucose will be broken. Of course, there's a whole bunch of the green proteins and a whole bunch of the next protein as well. So this is the chemistry of life, at least one aspect of the chemistry of life. The cell is making many different kinds of proteins and many of each kind of protein to achieve the goal of the breakdown of glucose. Now, to stay alive, cells must continually build new molecules, which requires ATP energy. So here we have the scene where this protein is linking together a couple of molecules, but that requires a source of energy. ATP carries that energy. But you might be asking, well, why doesn't the cell just use glucose as a source of energy to power the chemistry? Why does the cell go through this process using these proteins up here to package the energy of glucose into ATP? Why not just use the energy of glucose directly to power the chemistry of the cell? Now, to answer that question, let's look more closely at the protein doing the chemistry here. So this protein's job is to link the red molecule and the green molecule with a chemical bond. Now, that process requires energy. Now, we're going to use a financial analogy here and say that the amount of energy this protein needs to do its job is $1 worth of energy. But here's the situation. Glucose has $36 worth of energy stored in all those chemical bonds. So this is the problem that the cell faces. It's not appropriate to use glucose to directly power this chemistry. The chemical reactions require smaller quantities of energy to proceed. So the solution that cells have evolved is the ability to convert the $36 of energy in glucose into 36 molecules of ATP, each with $1 worth of energy. And we're going to call these proteins up here the bank proteins, because it's their job to do this kind of energy conversion, converting the energy in glucose, $36 worth of energy, and repackaging that energy into 36 molecules of ATP, each of which has $1 worth of energy. Now, this chemistry is very complex, and the people who figured this process up here out, they won Nobel Prizes. But so we're not going to go into that in this course. We just want the general concept that what cells are doing is that the energy of glucose is going to be stored in the chemical bonds of the ATP molecule. So each ATP molecule will have a fraction of the energy stored in glucose. Now, ATP has this structure. It's called adenosine triphosphate. So it's got like one of the DNA bases here, a ribose sugar, and it's got these 3 phosphate groups. And the bond between the second and third phosphates, that's this high energy bond that stores $1 worth of energy. So a fraction of the energy of glucose is being stored in that chemical bond right there. Again, if we think glucose has $36 of energy, that bond right there has $1 worth of energy. And that's exactly the amount of energy that lots of proteins need to perform their chemical reactions. So the proteins that make up the bank of the cell convert the energy stored in glucose, $36, into the energy stored in ATP. But it's many molecules of ATP, each having $1 worth of energy. So all known cells on planet Earth use ATP to power their chemistry. This seems to be a universal feature of life on Earth. Now, cells must build the molecules out of which they are made using ATP energy. So now it's time to examine this scene down here. How are cells going to be using ATP energy to build various molecules? We'll pick that up in the next part.